We have more people. All right, good afternoon, everybody. Thank you for coming to our third 3MT competition. For those of you who haven't been here before, the 3-minute thesis competition was founded by the University of Queensland in Australia with the goal of having students present their research in 3 minutes for a non-specialist audience. So for our competition, presentations are limited to 3 minutes for scoring. I will stand up. I will be down in the front by Catherine. I will stand up when you've reached 2 minutes and 30 seconds to let you know you have 30 seconds left to be scored. If you are over 3 minutes, you are disqualified from the competition. I will advance your slide at 3 minutes and 30 seconds and officially cut you off the stage. You're only allowed one single static slide. There can be no transitions, animations, or movements of any kind. You will be disqualified. You can have no other additional media or props, with the exception of some speakers have a paper with some notes to help them. The presentations are to be spoken. Dr. Coyne wants me to stress that you are not to rap or sing because they are different, and you cannot have poetry. We do have People's Choice Awards. The students are being judged by our 3 judges, which are Dr. Barrett, Dr. Matoka, and Dr. Poffenberger. But you as the audience have the opportunity to vote for the best speaker. There's 2 slips in front. One is blue, that's for the best student. And then the slip that is green is for the best faculty, staff, postdoc, or intern presenter. Please vote only once per category, and those will be due at the end of the event. So while we are playing today, to give the judges some extra time, we will be playing trivia between the rounds. So to get started, if you'll text CarryNot 557 to 37607 on your cell phone, you can participate in interactive trivia, and we'll have a practice question as soon as everybody texts their username in.
You gotta repeat the number for me.
All right. Everybody ready or no? Are we okay now? Yes. All right, so to practice, what is Dr. McGrath's Twitter handle? Is it NPK man, NPK professor, NPK boss, or NPK educator? And you just text A, B, C, or D. You good? All right, Dr. McGrath tweets as NPK professor. When we take a break for lunch, he's supposed to tweet us a special message that we will reveal at lunch when we come back. So to start, Dan Quinn, a PhD student with Dr. Chad Lee, will begin.
So the use of a rye cover crop, typically planted following fall soybean harvest prior to spring corn planting, continues to be of interest to Kentucky corn farmers. And this is due to rye's ability to reduce weed populations, as well as limit soil erosion, soil nitrate losses, and also improve soil organic matter and environmental quality. However, when it comes to the implementation of proposed beneficial farming practices, there's often challenges that arise. And with those challenges, a lot of farmers end up seeing yield losses when using rye, and that oftentimes limits their adoption of using rye. And some of these challenges can be illustrated by the two pictures above. These pictures were taken from side-by-side research plots from one of my Lexington research trials this year. Corn pictured here on the left was planted following a rye cover crop. The corn pictured over here on the right was planted following no rye cover crop. Corn on your left illustrates, exhibits nitrogen stress as well as decreased growth compared to the corn illustrated on the right. And this, in addition to the decreased growth and nitrogen stress data across 4 site years and 3 Kentucky locations, we observed corn following rye requires upwards of 22 pounds of nitrogen per acre more, as well as 7,000 seeds per acre more in order to maximize grain yield. And these increased requirements of corn following rye is often due to increased nitrogen mobilization, as well as increased nitrogen volatilization, as well as decreased corn seedling emergence, which is likely a function of increased corn seedling disease, as well as increased pest pressure when corn is following rye. So the goal of my research trials are not only to try and understand and quantify these seeding rate and nitrogen rate differences that we observe when corn is following rye or no cover crop, is that we're also trying to understand which management practices may also need to be implemented or adjusted in order to decrease this gap between the nitrogen rate and the seeding rate. And some of those management practices include starter fertilizer use, include in-furrow fungicide use, as well as adjusting nitrogen fertilizer application timings. So this research trial and other research trials I have hope to provide farmers with valuable tools so that they are able to, 1, decrease profit and yield losses when following a rye cover crop, and 2, be able to allow more farmers to implement the use of a rye cover crop so that they may be able to capitalize on proven soil quality and environmental benefits when choosing to use a winter cover crop. Thank you.
Our first trivia question of this round: what was the first college in the area that would become the Commonwealth of Kentucky? Was it the University of Kentucky, Kentucky State, Center College, or Transylvania University? All right, the first college was Transylvania University. It was named for the colony of Transylvania, which meant walk through the woods, and it was founded in 1780 when the land that was what would become Kentucky was still part of the colony of Virginia. At its founding, it was the first college west of the Appalachian Mountains. It was originally housed in a log cabin in Boyle County, Kentucky, and was moved to Lexington in 1789 and officially became a university in 1799. So our next question is, what was the first color scheme for the University of Kentucky? White and blue, gray and blue, white and yellow, or yellow and blue? Yellow and blue was the original color selected in 1891. The color of blue that was adopted was picked off of the tie of one of the football players. So for Dr. Coyne, that's why ties are important. The next year, the yellow color was dropped and it officially became blue and white. Our next speaker is Hannah Somerville, who is a master's student with Dr. Chris Shepard.
Okay. So when I talk about soil judging, I usually get two questions. First, what in the world is that? And second, why in the world would you do it? And the answer to both is soil judging is awesome. That's the what and the why. I can't think of another activity that enhances the undergraduate experience the way that soil judging does. Not only do soil judgers get the opportunity to gain skills and field research that they can take to any career in the environmental sciences that they may go to, but they also get the unique opportunity to network with both state and federal agencies like NRCS and USDA. They also get the opportunity to network with dozens of other schools that attend the soil judging competitions every year. But what is soil judging? Well, other than being awesome, soil judging is a collegiate version of FFA's land judging program. Both of those programs were created with the vision that we can use soil's morphology characteristics to determine how well that soil is suited for agriculture. So before the week of the contest, the host school's pedologist and the state soil scientist will go to the field and they'll excavate several pits, which you can see here. And those pits will contain soils that are either unique to or characteristic of the host school's region. A team of professional soil scientists will then do a morphological description of the pit, and that morphological description is what our soil judgers try to match whenever they enter the pit during the competition. But what goes into that description? So during the contest, soil judgers have 50 minutes to describe the morphology of up to 6 horizons within the pit. And the morphology of each horizon includes structure of that soil, color, texture, clay percentage, rock fragments, among other things. And then they'll use that morphology that they've determined from inside of the pit to make choices about the land's capability to be used for agriculture and some engineering interpretations. They also have to determine the taxonomic class for that soil. So that sounds like a lot, and it is a lot, especially for 50 minutes, but the apt undergrad can get really good at it with just a couple days of practice. This year, University of Kentucky competed in the Southeast Regional Soil Judging Competition for the first time in about a decade with our new coach, Dr. Chris Shepard. Our first-time team placed 6th out of 13 competing teams, and those teams were already established, so you know they're real good. We also had 2 soil judges place in the top 20, with Jeffrey McCall coming in 11th place and Morgan Vogt placing 14th. So to wrap up, though this was UK's first year competing in the soil judging competition, our soil judgers are a force to be reckoned with. Though through the contest, they became better environmental scientists, and they'll be able to take those skills with them wherever they end up in their careers. And, you know, I'm sure if you asked Jeffrey, Morgan, Nat, or Rebecca, they would all agree with me that soil judging is awesome. Thank you.
Memorial Hall is dedicated to Kentuckians who died in service during which war? The Civil War, World War I, World War II, or the Korean War? Memorial Hall was completed in 1929 as a memorial to those who died in World War I. Cases within its central hallway display the names of all students who served in both World War I and World War II from all counties within Kentucky. The University of Kentucky established the first extension program in 1910. What act of Congress made extension services mandatory at all land-grant institutions? The Morrill Act, the Hatch Act, Smith-Lever Act, or the Smith-Hughes Act? It was the Smith-Lever Act. So the Hatch Act of 1887 expanded the land-grant mission by establishing agricultural experiment stations at all land-grant institutions, but the Smith-Lever Act required that there be a cooperative extension service. Because UK did found or have the first cooperative extension service, it served as the model for all other land-grant institutions after the passage of this act. Our third speaker will be Dr. Bruce Downey.
So hello, I'm a seed biologist, and I'm here to talk to you a little bit about seeds. You might think you know about seeds, you don't. Here I am telling you about seeds. I'm going to speak specifically about orthodox seeds, those that actually have built in a program to dehydrate down to very low levels right after they are abscised from the mother plant. So on this axis, I have the percent of water, fresh weight. On this axis, I have time during the latter stages of seed development, the quiescent period, and germination, sensu stricto, followed by seedling establishment. If you look at water, you can see this dip and then this dehydrated period. When the seed finally imbibes, it gets water and continues on until seedling establishment. We call this particular period the latter stages of seed development, and it's in this particular period that the protective mechanisms and the repair mechanisms are actually produced, both proteins and RNA. So those protective mechanisms are actually functional during this dry period, which is a period of suffering where no synthesis or repair can actually take place. But what you've put in place already is protecting your internal mechanisms, if you will, proteins, membranes, etc., from damage. After imbibition, sensu stricto, when the seed imbibes and takes up water, that's the commencement of seed germination. And that continues through the lag phase of water uptake, so imbibition, lag phase, and a little period of embryo expansion until such time as the embryo protrudes from the seeds. Once that happens, that's a completion of germination, and now we're talking about seedling establishment. I focus my research both on this period and on this period, protection and repair. If you look here, we have the stored RNA and protein production being commenced in this period, and no synthesis or repair during this period, and then the existing proteins that you made over here are utilized here. The RNA that you made here is also utilized, and proteins are synthesized from that stored RNA to actually get you through this period. And then finally, you start to make new RNA from which you can also transcribe or translate proteins. This particular period here can be interrupted by a stress, particularly dehydration. And that is when you can also invoke this protective and repair mechanism a second time to get you through to the point of seedling establishment. Thank you for your attention.
William T. Young, for who the Maine Library is named, made his fortune producing what food item? Peanut butter, grits, molasses, or bourbon? So it was peanut butter. His company, WT Young Foods, manufactured the Big Top peanut butter brand. His business was one of the leading producers of peanut butter in the U.S., and he sold it to Procter & Gamble in 1955 when it was rebranded as Jif. And our last question, which Kentucky county was a dry county until 2016? Woodford County, Estill County, Bourbon County, sorry, Andrea, or Jessamine County? Does that work?
I said it.
I was like, Andrea always does.
I think you could, didn't I?
So it was Bourbon County. Our fourth speaker in this session is Andrea Keeney. She's a PhD student with doctors Andy Bailey and Emily Pfeiffer.
Good afternoon, everyone. Thank you, Rebecca, for the introduction. Like she said, I am a PhD student here in the Plant and Soil Science Department, but I'm going to be talking to you all about the key results from my master's project, which I just finished last semester. So many of you have heard me talk about this quite a few times over the past 3 years, but I worked with tobacco, potassium, and tobacco-specific nitrosamines, or TSNAs. Some TSNAs are known carcinogens. So with this project, I used 2 different potassium sources, potassium sulfate and potassium chloride. I used those over 3 different rates, ranging from 112 kilograms of potassium per hectare up to 336 kilograms of potassium per hectare. With this project, I did use 3 different tobacco types. Those were dark air cured, dark fire cured, and burley tobacco. I also had 3 locations. 2 of those locations were in Western Kentucky, 1 at the research station in Princeton, and then 1 at Murray State University. My burley trial was here in Lexington at Spindletop, and these trials were conducted over a 2-year time span. So with all of that, that gives us 12 trials that we did conduct data over. So with the results that we found that we had a significant yield response to potassium in 5 of our 12 trials that we had. So all of our initial soil index for all of these trials and for all of these years was less than or equal to 150 kilograms of potassium per hectare. Further results showed that we only had 1 negative effect of potassium chloride on cured moisture content in 1 of 12 trials. That was very surprising to us because there has been literature shown that as you increase your rate of potassium chloride, you can also increase your moisture content in your cured leaf. Also, more specifically about potassium chloride, farmers are regulated from applying no more than 110 pounds of potassium chloride, or 110 kilograms of potassium chloride per hectare. And that is due to moisture issues and also issues during the curing process, such as grade quality index, combustibility, and a bad aroma in combustible products such as cigarettes. So more specifically, looking at the quality grade index, so to us, it was very, very surprising, and we checked this data multiple times, but we had no negative effects of potassium chloride on the quality grade index in any of our trials that we had. But our biggest take home from this project is that we had a 30% reduction in TSNA from where we applied potassium chloride across all of our trials. So with all of these data points, R.J. Reynolds Tobacco Company funded this project. They have taken this project and they are doing it on a large scale basis to see if these points hold true on a larger scale. And with that, thank you.
What is the official bourbon of the Kentucky Derby? Woodford Reserve, Maker's Mark, Four Roses, or Wild Turkey? In a contract agreement with Churchill Downs, Woodford Reserve is listed as the official bourbon of the Kentucky Derby, is used in all of the $1,000 mint juleps sold on Derby Day. A special commemorative bottle is released for each Derby, and its sister brand, Old Forester, is used to make the moderately priced mint juleps. And the last question for this round, which Kentucky county has the most fertile soil in the state? And I'm not reading them this time. This is according to the internet.
Always right.
Yes, always.
How are they defined?
I couldn't bring up the internet map, sorry. Oh, there it is. Well, it's Barron County. And now we'll have Sam Leuthold, who's a master's student with Dr. Hannah Poffenberger.
Great. Thanks, Rebecca. I'm Sam Leuthold. I'm a first-year master's student at Poffenberger Agroecological Nutrient Cycling Lab. Today, I'm going to talk a little bit about my thesis work and the results of our first field season's data. So, large swaths of agricultural production land across Kentucky and most of the southeastern United States are characterized by rolling hill topography. This complex agricultural landscape leads to increased management challenges for producers as a downslope movement of fertile topsoil, soil water, soil nutrients create a mosaic of interspersed low fertility and high fertility areas throughout the landscape. This effect is then exacerbated as areas of different fertility produce different amounts of residue, leading to different soil organic matter stocks, reinforcing this landscape variability in soil fertility. So my thesis work really aims to address this problem, asking the question, can we use cover crops to mitigate some of this spatial variability and increase corn yields stability? To that end, I have 3 cover crop treatments, a cereal rye crimson clover mixture, and a winter fallow spread across 3 landscape positions, a summit, a back slope, and a toe slope. And to understand the effect of these cover crops on spatial variability, first we looked at the effect of spatial variability on the cover crops. And we can see that in this top plot. As we move left to right across the x-axis, we're moving down slope, summit, back slope, toe slope. We have above ground biomass of our cover crops on the y-axis. We can see a significant landscape effect on cover crop biomass with our toe slope area, which we would think to be the more fertile area, producing more biomass than either of our less fertile areas, the more eroded areas, the back slope and the summit. Interestingly, we also see that the mixture produces more cover crop biomass compared to the rye monoculture alone, regardless of landscape position, without any sort of interactive effect. So finally, in this bottom plot, we can finally answer this question that I've posed. Do cover crops mitigate some of this spatial variability in corn yields? And you can see that in both a nitrogen limited and a non-nitrogen limited setting, landscape position is still a major driver of corn yields. We did not see a significant effect of cover crop, and landscape position was the real driver of corn yields. So anyway, cover crops aren't a silver bullet. 1 year of cover cropping is not going to solve spatial variability, but it's important to continue this study and studies like it so we can better answer this question in the future. Thank you.
What was the only racehorse to beat Man o' War? Upset, Blazes, Garbage, or Whisk Broom the Second? So it was the horse named Upset. Upset and Man of War raced 4 times, with Man of War winning 3 out of the 4 races. And he overall won 20 of his 21 races, including a race against Sir Barton, who would become the first winner of the American Triple Crown in 1919. And another horse question: what is the only Triple Crown winner to sire another Triple Crown winner? American Pharoah, Secretariat, Gallant Fox, or Citation? So it was Gallant Fox. Gallant Fox was the second Triple Crown winner in 1930. Omaha, his offspring, was the third Triple Crown winner in 1935. Our sixth speaker for the 3MT is Lucia Garcia Vidal. She's an intern in the Rhizosphere Science Lab.
That's Argentina, my home country. And that 5 spots in the map represent the farms of my family company, Familia Garcia Vidal. In 4 of these farms, we have a livestock production system. In Taco Pozo and Fontana, we do cow-calf operation, where we separate the cows with 160 kilos. Taco Pozo has very arid conditions as a result of very low annual precipitations and the presence of sandy soils. Fontana, on the other hand, doubles the amount of annual precipitations and presents heavy forests and natural pastures. If you follow the arrows, you'll see that the animals then go to Chorotis, and they are going to stay there until they reach 300 kilos. The picture illustrates Gaton Panic, a natural pasture that grows in the region. Finally, the animals will go to Entre Rios, and the process will finish when they reach 460 kilos. This farm is located over the Paraná River. This is the 2nd largest river of South America after the Amazon River. Food here abounds as long as the water stays between certain levels. If the river rises too much, then the island will get flooded and the animals will have to be removed until the water goes down again. That explains the arrows that show that the animals go to Pergamino and then go back to Entre Rios. Why do we choose to finish our cattle in Entre Rios? That is why in that region you'll find all the big companies that will buy the animals and export the meat to the rest of the world. The breed of cattle we use is called Senepol, and you'll see that all the cattle that we produce is grass-fed only. That gives special characteristics to the meat regarding flavor and tenderness. Last but not least, in Pergamino, the 3 pictures on the bottom, we grow crops and raise pigs. The crops that we do are soybean, corn, sorghum, and barley. Part of that crop grains is sold to an agricultural cooperative we work with, and the rest of the crops, of the grain crops, will be stored in our silos located in the farm, and it will be used to feed our pig production. As you can see, our goal is to integrate all of the farms and produce food for the rest of the world. And well, finally, that picture is me with my father in one of the farms. That's my father and founder of the company. Thank you very much.
How many public 4-year institutions are in Kentucky? 6, 8, 12, or 15? There are 8 4-year public institutions. They are the University of Kentucky, Murray State University, Morehead State University, Kentucky State University, Eastern Kentucky University, Northern Kentucky University, Western Kentucky University, and the University of Louisville. Lastly, farmland occupies the greatest percentage of what state? California, Iowa, Nebraska, or Texas? %93 of the acreage in Nebraska is classified as farmland, whereas Texas has the most acres of farmland and California produces the most food of any state. Our next speaker is Dr. Olga, and she'll be speaking about nanoparticles.
I see. No? Okay.
You had
Works.
the laser pointer earlier.
No, it's working. It's working. It's working. Verify. Okay. Applications of gold nanoparticles in medicine are rapidly increasing. Due to their physical-chemical properties, they're being used in imaging for diagnostic purposes and also in drug delivery for therapy. In drug delivery, they're being considered to be used to deliver drugs to the brain for treatment of neurodegenerative diseases. There are a lot of research done on the toxicity of gold nanoparticles, often with contradictory results. Meaning a lot of the studies show that gold nanoparticles can act as Mr. Hyde, which is a bad guy. What do I mean by that? I mean that when gold nanoparticles enter cell, they can activate a lot stress response mechanisms, including endoplasmic reticulum stress, unfolded protein response, they can cause mitochondrial dysfunction, all of this leading to apoptosis or cell death, which is a bad thing, right? So can we tune down those effects of Mr. Hyde and make gold nanoparticles sort of act as Dr. Jekyll, which is a good guy right there? Well, there is a hope. So our studies are being done in model organism, C. elegans, and this model organism has been used for the last 2 decades as for fundamental research that is done in aging. And our study demonstrates that if you induce only mild stress when you expose nematodes to gold nanoparticles, you actually can activate longevity mechanism instead of cell death. And activation of this longevity mechanism occur by induction of 2 specific conserved transcription factors. One of these transcription factors, HLH30, or it's called TFAP in mammalian cells, is key regulator of autophagy. And those transcription factors work together to accelerate autophagy and promote longevity. What is autophagy? Autophagy is evolutionarily conserved mechanism, cleanup mechanism of degradation of protein aggregates and cell debris. And as you know, many neurodegenerative diseases have the common feature is accumulation of protein aggregates, right? So regulation of autophagy and specifically enhancing of autophagy serve as target when developing novel potential treatment of neurodegenerative diseases. So if we can use gold nanoparticles as potential means for enhancing autophagy, as well as for identifying novel autophagy genes, that actually will help with the treatment of neurodegenerative diseases. Thank you.
Who applied the Munsell color system to soil and standardized the methodology for determining soil color? Albert Munsell, Hans Jennings, Hugh Bennett, or Dorothy Nickerson? Dorothy Nickerson worked at Albert Munsell's company, named the Munsell Color Company, as a laboratory assistant and a secretary for his son. She then transferred to the USDA in 1927 and applied color theory to agriculture for the first time. She developed a color chart for horticulture, which is still used today, known as the Nickerson Color Fan, and she arranged the Munsell Color Book we use for soils. She was the author or co-author on 150 publications related to color and its application in agriculture. What two soil orders are formed in organic materials? Histosols and mollisols, mollisols and alphasols, spodosols and alphasols, or histosols and alphasols? They are histosols and mollisols. Mollisols cover 7% of ice-free land, whereas histosols cover only 1% of ice-free land. Our next speaker is Mohammad Fateh Ali. He's a PhD candidate with Dr. Tomo.
Before I start, I would like to take you to your imaginary world. I would like to ask you to think about a world without plants. What you will see? You will see probably something like mud. Yes, basically, if there is no seed, that means no plant. No plant, no food. No food means no human being. So in flowering plants, seed development is followed by pollen tube. Followed by pollen tube guidance, where the pollen tube contains two sperm cells, one of them fertilized with the egg cell to generate embryo, another one fertilized with the central cell to generate endosperm. And today, during my talk, I will mostly talk about the endosperm development, which is the major part of my PhD thesis. Endosperm actually serves as a nourishing tissue for embryo development, and it's also the major source of food for human beings. And during the seed development in some crop like corn, wheat, or rice, the part that we eat for our food is actually the endosperm. On the other hand, some crops like here, the soybean, here the embryo actually consumes the endosperm during its development. From Arabidopsis research, we have extensively studied the embryo and endosperm development. However, when it comes for soybean, which seeds is much larger, it was not easy to begin with. However, I have developed a new method to study the embryo and endosperm development in soybean without doing any sectioning. In soybean, actually endosperm followed a liquid phase of development. Here you can see in this image the endosperm actually liquid endosperm, where the central vacuole of endosperm large rapidly, and the central vac in this vacuole actually the nuclei of endosperm divides without any cytokinesis. And after certain division, in the right image here you can see this embryo, and after certain division of nuclei the endosperm start to cellularize. Here actually the cellularized endosperm. And from Arabidopsis research, we also know that if there is any disturbance of the timing of this endosperm cellularization, it can cause a small or aborted seed. And yes, I am applying this knowledge from my Arabidopsis to the soybean project. So in soybean project, now I am applying the temperature treatment, and with this treatment, I want to observe the embryo and endosperm development, which is my major interest, and also endosperm as the major important factor to determine the final seed size or yield in case of soybean. Thank you.
True or false, Kentucky does not have any soil classified as a vertisol. So it is false. The Sharkey Soil Series is the only vertisol in the state of Kentucky. It is a clay alluvium on the Mississippi River floodplain and is predominantly found in Louisiana and Arkansas. Kentucky was the site of the first commercial what in the United States? Bourbon distillery, whiskey distillery, winery, or distilling barrel manufacturer? C, we were the site of the first commercial winery in the United States, aptly named First Vineyard in 1799. Our next speaker is Annie McGraw, a master's student with Dr. Chris Shepard.
$11.5 million square miles. That's the area of the Earth's land surface that is considered karst. That's roughly 20% of all dry land. So what is karst? Karst is a cool type of topography that forms as a result of the dissolving of certain bedrocks. More specifically, rain coming down, water coming down as rain, reacts with the carbon dioxide in the atmosphere to form carbonic acid, which dissolves the calcium carbonate in rocks like limestone, which is very common here in Kentucky. This process results in a variety of both surface and subsurface features. Sinkholes are the poster children of the karst landscape, but most people tend to think of cover collapse sinkholes. Think of what happened in 2014 at the National Corvette Museum in Bowling Green. That was cover collapse, but not all sinkholes are so flashy. My research looks at the soils and cover subsidence sinkholes. As this graphic up top shows, they're not nearly as dramatic as the cover collapse. They're more of a slow sinking of the ground surface above only partially dissolved bedrock. Because karst is a result of such rapid processes, we are able to see changes in these features within our own lifetimes. The soils in these sinkholes are what interests me the most. The carbon cycle, shown here, is just that it's a cycle. The ability of soils to hold on to this carbon and not release it back into the atmosphere is very important to the climate change narrative as a whole. If we can figure out where within sinkholes carbon is being stored and how much, we will be able to determine which areas need to be protected so they can hold on to this carbon as best they can. To do this, we need to sample soils from a large number of sinkholes. Once we have selected a sinkhole to study, we need to pick where along the side or hill slope to sample. Two interesting points for comparison are at the top, or summit SU, and at the bottom, orange toe slope. By comparing trends in the amount of carbon at these different landscape positions, and also comparing trends in the depth and level of carbon at these different depths, we will be able to get a better picture of how these features are storing carbon and how we can keep this carbon in the ground so it's not coming back up into the atmosphere. By finding out all we can about the soils in these karst systems and uncovering any trends in carbon storage, we will be able to appreciate their contribution to the global carbon cycle. I hope to bring more awareness to the value of these sinkhole soils by illustrating their importance in the global carbon cycle and also demonstrating the variability of these kind of wacky systems. And seemingly, even though they're super common in Kentucky, there's not a whole lot known about them, so hopefully we can demonstrate the value they're bringing, and because they are 20% of Earth's land surface and they deserve more than a little bit of attention. So that's it. Thank you.
Bourbon barrels are made exclusively from what tree? American white oak, northern red oak, pin oak, or black oak? It is American white oak. It was selected for its physical strength in making the cask, as well as some of the chemical properties that it can impart to the bourbon. The first franchised Kentucky Fried Chicken was opened in what state? Kentucky, North Carolina, Utah, or Indiana? The first Kentucky Fried Chicken was located in Louisville, but the first franchise location opened in North Salt Lake City, Utah at Harmon's Cafe. We'd like to welcome Madison Kramer, a master's student with Dr. Travis Legletter.
All right. Thank you for the introduction, Rebecca. Again, my name is Madison Kramer. I'm a second-year master's student working under Travis Legletter. Some of you may look at this picture and be like, what the heck are they doing? And some of you might be thinking, I haven't seen one of those be used in action in a long time. So weed wiper technology was mainly used in glyphosate-resistant cropping systems such as soybean. But now we've kind of shifted into the area of glyphosate-resistant weeds. We kind of have to go to different forms of herbicides to control problematic weeds such as Palmer amaranth and water hemp. So when you look at a dicamba label and you're trying to use a weed wiper, the instructions are really vague. So what we did is we conducted a study in Princeton, Kentucky, looking at how to calculate how much spray solution is actually being deposited onto a dicamba tolerant soybean system, and trying to looking at the overall efficacy of what a weed wiper if we're actually controlling weeds. So what we did is this is a 5-foot weed wiper. We filled it up with 400 milliliters of dicamba and water. So that's a 1-to-1 solution. So if you kind of think about that, 200 milliliters of water, 200 mils of dicamba, that's a lot. So what we did is we primed this weed wiper. So we went over a bulk area of dicamba tolerant soybeans, and we would prime it to make sure all those rope wicks were absorbed with the spray solution. So we had 2 different forms of methods for application. That first application is just a 1-pass. So we just walked down the plot 1 time, and then we did a 2-pass. So we walked down the plot 1 time, then reversed it and came back. So we had 3 different treatment styles we looked at. So the first treatment, we used a pre-emergence herbicide where we just touched the soybean, or we touched the soybean foliage and the weeds in the plot. And the second one, we did not use a pre-emergence application, and we strictly just touched the weeds. In this case, it's going to be water hemp. And we did not touch the soybean plant. And then the third application was a mimic of an escape situation. So when you come in late post and you have a couple of stragglers out above the soybean canopy, so we did that when the weeds were taller than soybean canopy. So some of the results suggest that we did 14 days and 21 days visualizations after application. The results suggest that a 1-pass or a 2-pass application is better than a 1-pass application, which is something we would expect. But the calculations suggest that there are very high amounts of dicamba being used when we use a weed wiper. So overall, I think more research needs to be done using weed wiper technology, but it is a good tool that we could use when we need to use dicamba in an area where we have a specialty crop being grown next door and in buffer zones. With that, thank you.
What is the other land-grant college in Kentucky? Morehead State, Kentucky State, Eastern Kentucky, or Northern Kentucky University? Kentucky State was established in 1886 and was the second state-supported institute of higher education, and it was granted land grant status in 1890. Now, if you paid attention to Annie, you know the answer. The only assembly plant in the world to manufacture the Chevrolet Corvette is located in which Kentucky city? Georgetown, Versailles, Lexington, or Bowling Green? It is Bowling Green, and apparently they had a sinkhole, and I didn't know about it. Our next speaker is Stu Lichtenberg. He's a PhD candidate with Dr. Jason Unrein.
So good afternoon. RNA interference is a cellular mechanism possessed by eukaryotes that allows for the selective silencing of genes. This is achieved through the introduction of double-stranded RNA. This double-stranded RNA serves as a template, which is used by these endogenous cellular processes to degrade a corresponding messenger RNA. Now, RNA interference has been used as a valuable laboratory tool for many years now, but other applications are starting to come to the forefront, particularly that of crop protection. Let's say, for instance, we want to control a given insect pest. We can use a double-stranded RNA that corresponds to an essential gene in a given insect species, and therefore use that double-stranded RNA for control of that insect pest. However, there are some notable pitfalls associated with the use of RNA interference as a pest control agent. Foremost among these is that double-stranded RNA degrades extremely rapidly in the environment. Estimates are that the half-life of double-stranded RNA in agricultural soils is somewhere on the order of 24 to 48 hours. Furthermore, many insect species do not readily take up double-stranded RNA alone. And lastly, because of the specificity of RNA interference, new constructs are required for each individual insect pest. In addition, the most popular means of applying double-stranded RNA as a pesticide is the use of transgenic constructs. Numerous crop species are not amenable to genetic transformation, and in many areas are regulated to the extent that they're not usable in those countries. So what is a solution for this? There's a single technology that can overcome many of these limitations, and that is nanotechnology. By combining double-stranded RNA that's synthesized in vitro with nanomaterials, we can overcome many of these problems. And that's what my work has focused on for the past 4 years. Using the nematode C. elegans, seen at the center right here, we've been able to conclusively show that double-stranded RNA nanomaterial conjugates are assimilated into cells in ways that are distinct from the traditionally understood means of naked double-stranded RNA uptake. Furthermore, we've been able to identify a number of environmental factors that strongly impact the efficacy of these particles, including pH and the presence of natural organic matter in soil solution. Our work is going to serve as a platform for developing the safest, most specific pesticides ever produced, and truly see realization of Rachel Carson's dream of an ideal pesticide. Thank you very much.
As our judges finish scoring, we have 2 more questions before lunch. So many Kentucky cities are named for European cities. Which of the following is not a city in Kentucky? Athens, Glasgow, Paris, or London? Athens is not a city in Kentucky. There is a Paris, London, and Glasgow. Glasgow has a Scottish Highland Festival every year if you would like to attend. Lastly, Nick Wilson is a UK alum who won which reality TV show in 2018? The Voice, The Amazing Race, Big Brother, or Survivor? He won Survivor season David versus Goliath. He's a public defender from Williamsburg, Kentucky, and was a first-generation college student at UK who went on to earn his law degree at Alabama. He famously formed an alliance with a player from Maryland that they named Team Mason-Dixon. So it's time for lunch now. We have lunch from Johnny Carino's with pies from Missy's Pies. We'll reconvene at 2 o'clock. You know, I gotta do my best to make things funny. You got the slides down there. Well, we got 2 advertisements. All right, welcome. All right, welcome back, everybody. So before we get started, we have 2 advertisements because what's an event without advertisements? So the first is the GSA has polo shirts for sale. For graduate students, we've received money from the Graduate Student Congress to put towards the purchase of the shirts. So you will place a pre-order with Audrey and Abby, and then we will tell you the price once we know how many shirts we have to order. For faculty and staff, it's just the $25 or the $27 or $28. We have both a men's and women's cut available in blue and gray, and they'll have the new UK Ag logo, but instead of extension, it'll say Plant and Soil Sciences. And then lastly, we also, courtesy of Dan, have made mugs. So instead of the white mugs in the back, you can have your own Plant and Soil Science mug. And this one is a fundraiser for our professional development programming. And I am a couple minutes ahead of schedule. And I don't have more trivia. Oh, good. Yeah, I was gonna say, Dr. Matoka, you got a joke? I think, Dan, we had jokes earlier. Tell a joke. All right, Catherine's joke is y is 7? Y is 7 the scariest number? Yes. What do we have earlier? What did the buffalo say when he dropped his son off at college? Bye, son. How do you make a Kleenex dance? This is Catherine's joke. You put a little boogie in it. All right, we got 1 more minute for an audience joke. All right, and our first speaker in our afternoon session will be Norbert. He's a PhD student with Dr. Seth DeBolt.
I can just go whenever. Oh God, I'm so not ready. Okay, hi, I'm Norbert. I'm a PhD student working in the DeBolt lab. First, I'd like to thank the organizers of this lovely competition for putting it on for us. So thanks for that, dragging me up here. But I work, like I said, in the DeBolt lab with stock lodging. So what is stock lodging? Stock lodging is just a fancy way of saying This is what happens when a plant falls over before we're ready to harvest it. So why do we care about that? Well, it's the food we eat being left on the ground. And anywhere from 5 to 20% of annual grain yields are lost due to this phenomenon every year. So before I actually joined with this lab about a little under a year ago, the only exposure I've ever had to stock lodging was as an undergraduate student back at Mississippi State University in a maize genetics lab. And I remember every year we'd go out and our researcher would hand us a shovel, force us to walk through the mud and assess the damage that the previous night's storm had caused on our experiments. Best case scenario, if something had fallen over, it was root lodging. And we just pull the stock back up and pop a shovel full of mud up to just prop it up. Plant kind of sort of recovered, not really. However, if it's stock lodged, not the same equation here. Sometimes it was just crumpled at our feet on the ground, broken and useless. Other times, some would say even worse, it was fallen over onto one of its neighbors and negatively impacting its potential yield. So stock lodging, as I've come to know in the past year, is a very complex agricultural phenomenon that's caused by numerous environmental, genetic, and agricultural factors. In order to study this, we've pulled together some researchers from across the US. We've got researchers here at the UK, Monty's on it, so is Chad, researchers at the University of Idaho, and also at Clemson, and we've pooled their collective talents in everything from cell biology to genetics to a statistician on board to study the underlying intermediate phenotypes associated with stock lodging. The project is still in its relative infancy. However, we've made some serious progress in developing a new phenotyping tool that we can use to accurately and reliably measure the forces required to lodge maize and sorghum stalks in a way, and this is kind of important, that mimics natural stock lodging events, right? Over the past summer, myself and some other graduate students have had the opportunity to use these devices to start collecting some preliminary data points we can use to flesh out the compositional and morphological variables associated with and defining the strength of a stock. I'm almost out of time, so I work with corn and sorghum. We're looking at the strength of stocks, and we want to build models that can predict those strengths in a lot of environments. Thank you.
All right, we're back to trivia. Steamer milling provides the flour for which company's brownie mix? Ghirardelli, Duncan Hines, Betty Crocker, or Hershey? And if you were on the fall PD trip, you know the answer to this question. It is a Ghirardelli. So support Seymour Milling, who supports us, and get your Ghirardelli brownies. The UK wheat breeding group is currently working on what wheat disease? Stem rust, downy mildew, head scab, or leaf rust? It is head scab. The Fusarium pathogen results in significant yield losses and reduced grain quality, and it can produce a vomitoxin known as DON. Our next speaker is Dr. Travis Shaddix.
Okay, hi, I'm Travis Shaddix. What I wanted to do today was spend a little bit of time to walk through the entire process as I see it from what I call observation to impact. So in our world of turfgrass science, it's very common to use fertilizers that contain micronutrients, in this case particularly iron. The most common iron source in our world is iron sulfate, granular iron sulfate. But every time we pulled out that iron sulfate out of the material, and we've actually applied it to the turfgrass, I've never seen a response to that iron sulfate. In fact, we see the opposite. over in the top right-hand corner, when we see staining on concrete or impervious surfaces, that's a result of the iron sulfate oxidizing out into iron oxide, rendering it unavailable to the plant. So we generated a study, and our hypothesis was that granular iron increases soil-soluble iron. And it was a very, well, it was rather complex, very simple laboratory incubation study where we had 11 iron treatments. We had 11 soils from around the United States, including 6 different states and 7 different states and 6 different universities. We had 5 time levels where we applied the iron to the soil, and then we extracted the soluble iron remaining in the soil at each time level from 1 hour out to 21 days. And we used Glimmick's and 2B Kramer for the statistics. So you can see in the graph in the bottom middle that essentially this is a summation of all the soils and only looking at 1 hour and 21 days. And what it's saying is basically the chelated iron sources remain soluble. The non-chelated iron sources do not, including pretty much every granular iron source you can come up with that is not chelated. They're the same as untreated soil. So our conclusion was chelated iron increases soil-soluble iron, and we didn't find any evidence supporting the claim that non-chelated sources increase soluble iron in the soil. So what I would encourage people to consider at this point generally, and I've been guilty of this, is stopping our research at that point and we have a conclusion and we write our thesis and we put it in the library somewhere or whatever. But I'd really encourage people to consider pushing it forward, getting it published into a refereed publication, whatever that may be in your world, if it's the Tri-Society or Weed Science or whatever that might be, so that it's useful to people beyond who's going to read your thesis. However, even your thesis, even your refereed publication is only useful to those people who really read that, which is a very small portion of the population, right? So move it again further if we can, especially, and again, I've been guilty of this with students not getting their refereed publications out into extension pubs. You get it to extension pubs, whether it's trade journals or whether it's our own internal extension publication. And in that way, we can begin to use those resources through extension to make an impact, which in my definition is to increase or enhance the profitability of Kentucky enterprises or the well-being of humans if you're in the world of clinical health. Okay? That's it.
Former Wildcat Benny Snell now plays professional football with which NFL team? The Miami Dolphins, the Oakland Raiders, the Pittsburgh Steelers, or the Kansas City Chiefs? It is the Pittsburgh Steelers. He's number 24 there. Go ahead. All right, this Kentucky attraction is the only place in the Western Hemisphere in which a moonbow can regularly be seen. Natural Bridge, Red River Gorge, Bad Branch Falls State Nature Reserve, or Cumberland Falls. Oh, yes, state. It is Cumberland Falls. Moonbows are rare natural atmospheric phenomena that occurs when the moon's light is reflected and refracted off water droplets in the air. Moonbows are much fainter than rainbows made by the sun, and they often appear white. That's because a smaller amount of light is reflected from the surface of the moon. Our next speaker is Sarah. She's a PhD student with Dr. Travis Shaddix.
Good afternoon, everybody. My name is Sarah, and today my topic will be polymer-coated urea versus freeze. In another word, can polymer-coated urea survive during the winter? So polymer-coated urea, when you think about it, if you're not familiar with it, just think about a tea bag. So you put polymer coating outside the urea, and then which will allows water to gas in and bring out urea a little bit amount of the time, so which make it slow release. And here is a picture that I took after all of the urea are gone, the set little coating. So So the longevity of the polymer-coated urea depends on how thick the coating is. However, there are a lot of things can happen, like make the damage of the coating. Once the coating gets damaged, the urea will explode to the water, which make it not slow release anymore. So what will cause the damage? Well, there can be, that can happen in the transportation or handling, or even after you put it out, the human or the machine traffic can cause the damage. However, not many people think about if the temperature, if the weather can actually cause the damage. So what if you put it out during the fall and then it rains or you irrigate and the coating absorb the water and then the temperature drop? What if, will the water, after it turns into the ice, will it like actually cause the damage during this process? So so that's why we did this test run. We want to see if we can actually see any difference. Can you see any difference between these two pictures? Can you tell which one is the frozen or which one is not? Both of this one. What about this one? So actually this one is the freezing one. However, if you take a closer look, even though like I'm this close, you can't really see the big crack as we thought, like it will like crack out. So after this, what we think is we can test the difference of the nitrogen release, or we can actually use a special camera to actually record what will happen during the freezing period. And I think I end early, so thank you, everybody.
What was the first land-grant institution created under the Morrill Act? Michigan State University, Kansas State University, Iowa State, or Rutgers? So the first institution actually created after the passage of the Morrill Act was Kansas State in 1863. However, Michigan State and Penn State were granted land-grant status before the establishment of Kansas State, and so they are often called the first land grants. They have a commemorative stamp and a football game, Dan has informed me. However, Rutgers holds the title or the distinction of being the oldest current land-grant university with its founding in 1766 and later granting of land-grant status. Which of the universities below holds the distinction of being a land, sea, space, and sun grant institution? Oregon State, Ohio State, the University of Auburn, or the University of Delaware? There are only two institutions that are all for land grant, sea grant, space, and sun grant. That is Oregon State University and Penn State University. Our next speaker this afternoon is Uma Fatima. She is a PhD candidate with Dr. Tomo.
Hello, good afternoon. This is Uma Fatima from Dr. Tomokazu Kawashima's lab. Today I am here to secure your food source. As a graduate student from Department of Plant and Soil Sciences and from Seed Biology Lab, with no doubt I'm talking about seed. as a plant source food that directly or indirectly provides 60% of world total calories. But the matter of concern here, this 60% food supply is in danger due to adverse climatic conditions. This year, we all experienced severe drought in Kentucky, followed by a great reduction in overall crop yield. Onset of drought is injurious to plant health. And you know what? Among the life cycle and among the all stages in plant fertilization stage is the most sensitive stage. The problem here is we do not have control over environment. But if we know the underlying mechanism of plant fertilization, we would be able to manipulate it and assure successful fertilization even in that condition which is not normally favorable for plant. So this slide shows our knowledge about flowering plant fertilization with the future direction in compared to animal. Now you may ask, why in compared to animal? Because fertilization in flowering plant is unique and very different than that of animal. In case of animal, for successful fertilization, it only requires only one male gamete fused with only one female gamete. And in that case, female gamete nucleus migrate to male gamete nucleus with the help of cytoskeleton component microtubule. But in case of flowering plant, the scenario is different. It requires the fusion of two male gamete with two female gamete. And the migration of the male and female gamete nucleus is completely contrast to animals. Here, female gamete nucleus is static. And male gamete nucleus approaches to female gamete nucleus for successful fertilization. And in that case, they don't use microtubule. They use another cytoskeleton component, which is called filamentous actin or F-actin. But till now, we do not know how does this filamentous actin regulate sperm nuclear migration. Therefore, in our lab, using live cell confocal microscopy imaging with the combination of pharmacological and a genetic approach, we are trying to assess factors which is responsible for F-actin regulation during sperm nuclear migration using model plant Arabidopsis thaliana. Like, why I am doing this research? To fulfill your hunger so that you will not be starving in coming adverse climatic conditions, and can fulfill your hunger on my next 3-minute presentation. Thanks.
What vigilante group terrorized Kentucky farmers during the Black Patch Tobacco Wars for selling tobacco at too low of a price? The Night Riders, Morgan's Raiders, the Guthrie Riders, or the Black Patch Raiders? It was the Night Riders. I thought that was pretty good. What two Civil War figures were born in Kentucky 9 months apart? Jefferson Davis and Robert E. Lee, Robert E. Lee and Abraham Lincoln, Ulysses S. Grant and Robert E. Lee, or Abraham Lincoln and Jefferson Davis? Abraham Lincoln was born near Hodgenville. At the time of his election to the presidency, he resided in Illinois. Jefferson Davis was born in Fairview, less than 100 miles away from Lincoln's Hodgenville, and he studied at Transylvania University. Our next speaker is Rebecca Ramsey. She is a research analyst and lab manager in the Critical Zone Podology Lab.
So today I'm going to be talking to you about some work that I completed during my master's while attending the University of Alabama. And the plants and soils in question are those from coastal wetlands, and coastal wetlands are important. They provide habitat, economic products, and they're also the primary buffer for our coastlines against storms, waves, and flooding action. Now we need to cover a few main basics. Coastal wetlands maintain surface elevations through two main processes. You have physical and biological. Physical includes sediments being deposited onto the surface of your wetland through waves, storms, and tidal action. And biological includes the production and decomposition of the organic material that's within your wetland. Both of these processes add material to the surface of your wetland, increasing the elevation. However, if a wetland does not continue to add enough materials relative to the increase in sea level rise, which is continued and predicted to be increasing in time, they're at risk of being flooded out. So we lose all of those goods and services that the wetlands provide. Another basic is that coastal wetlands are notorious for their low oxygen content, and that'll be key a little later. With my project, I was supported by the University of Alabama and USGS to travel to the coast of Louisiana to collect 4-meter deep cores from 2 coastal wetlands. These coastal wetlands were radiocarbon dated from these cores to as much as 2,000 years before present. On the screen, you'll see a 0.5 meter core section, and within it, you'll see alternating bands of gray sedimentation indicated by these orange arrows and dark peat material. This is the evidence of those physical and biological processes that accumulate materials within your wetland. Within the peat material are vegetative fragments, so small bits and pieces of plants that used to be on the surface of the wetland before they were buried. Most keys are whole plant keys for identifying vegetation. For my project, I actually took live specimens that were on the surface of the wetland to cut them up into all the different bits and pieces that are possible to identify the bits and pieces that we found in our cores. On the bottom are just a few examples, and they're actually very unique, which makes the key easy to follow through. On the far right-hand side of the screen, you'll actually see the exact same species, but it's 2 different specimens. And the one on the top was collected from the surface, which was a live specimen. And the one on the bottom was actually collected at 80 centimeters depth within a core. So you can see that this live vegetation can identify your preserved vegetation only because it's preserved because of that low oxygen content within a wetland. So using the vegetation fragments that were identified, bulk density, percent organic matter, and radiocarbon dating throughout these entire cores, we're able to reconstruct a localized vegetation history for how both of these wetlands responded to past sea level rise, which gives us a glimpse in how they'll respond to future sea level rise and how we can best aid their preservation and preserve their goods and services.
What tree is most commonly struck by lightning? Willow, sycamore, redwood, or oak? It is oak. Oak tends to grow taller than most trees, and they have a lifespan of 300 to 600 years, which is why they believe it might be struck by lightning more. They also have a deep central root system and hollow water-filled cells that they say makes it a good conductor of electricity. Vanilla beans come from what flower? Orchids, roses, dahlias, or crocuses? Orchids are found in hot, wet, tropical climates, and most vanilla beans are harvested from orchids in Mexico and Madagascar. Our next speaker is Jared Cochran. He is a PhD student with Dr. Olga.
bacteria and then translate very well. All right, so antimicrobial resistance is a global concern, but what is it? And is it really a concern for you? Antimicrobials are a treatment for malaria, viruses, and bacteria. In the cases of bacteria, the antimicrobial is typically an antibiotic, and the specific resistance is known as antibiotic resistance. What's interesting and slightly frightening about bacteria is that they're able to house multiple genes which confer multiple versions of antibiotic-resistant genes. And they can spread these genes to other bacteria. This is the reason why when you're prescribed antibiotics, you need to finish the entire round, because if you don't lower the infectious bacteria population to a sufficient level, then the small fraction of antibiotic-resistant bacteria that may have developed or already been there can spread their genes to the remaining bacteria, and then now you have an infection with a head start and fewer treatment options. There are many sources for the development and spread of antibiotics. One such source is food production, as shown by this graph. Starting from the top and going counterclockwise, it shows antimicrobials have been overused by farmers to their livestock for decades, creating an environment that favors the development of antibiotic resistance. Once these bacteria develop, they can then spread to other livestock or to the farmer. And now through the mishandling of waste or through the farmer handling the food themselves, they can spread the bacteria to the produce. And then now your yearly salad can be a carrier for an infectious disease. I said there are multiple sources of antibiotic resistance, and they're all creating a world, a future where antibiotics may be ineffective. And everyday infections could prove deadly. This is a concern because our medical system is built on the reliance of antibiotics to manage infections. Without antibiotics, many medical procedures would no longer be tenable. Surgeries, for example, require antibiotics after the procedure. And without the antibiotics, it might be too risky to go through the procedure. Why have a hip replacement if the subsequent infection might prove deadly? Also, many other medical treatments increase the risk of an infection. Dialysis and cancer treatments greatly increase your infection. And again, it might just be too risky to go through it. You can't outweigh the benefits of an inevitable death. So what is to be done? Well, I'm working with Dr. Olga Tusko, and we're looking at an iron nanomaterial and seeing if it can treat an antibiotic-resistant bacteria using C. elegans as a model species, which I don't really have time to go into that procedure. But if we are successful, that might mean that antibiotic resistance might be a concern you no longer need to have.
Which of the following fruit was not native to North America? Cranberries, Concord grapes, apples, or blueberries? It is apples. Apples originated in modern-day Kazakhstan. Sweet dessert apples were domesticated in China, whereas firm and sour apples were domesticated in Europe. What is the #1 fruit crop globally? Bananas, apples, grapes, or oranges? It is bananas. On the bush, a single banana is called a finger, and each row of bananas is called a hand, and all the hands are called a bunch. So, in all, a hand usually has 20 fingers, and a bunch usually has 200 fingers. Our next speaker this afternoon is Dr. Jason Unrein.
Drinking water quality is a great opportunity for community participatory research. So many communities are rightly concerned with the quality of their drinking water and any health effects that they might have. Over the past year, I've been conducting a pilot community participatory research study in Martin County, Kentucky. My community partners for this study are Martin County Concerned Citizens, Kentucky Headwaters, and Appalachian Citizens Law Center. So during the 1970s, Martin County built up a pretty extensive municipal drinking water system. And that was necessitated by the depletion of groundwater from coal mining. Now that coal mining has subsided, there are fewer tax revenues and less resources to maintain the drinking water system. So as a result, it's in a poor state of repair, and they lose as much as 70% of the water that's produced due to leakage. This makes it hard to maintain adequate pressure in lines, so that leads to infiltration of soil-borne contaminants and an inability to flush out sediments and contaminants from lines, and subsequently biofilms accumulate in the pipes. And as a result of this, along with organic matter in the source water, there have been problems with formation of disinfection byproducts. The DBPs are formed when drinking water containing organic matter is chlorinated. So the DBPs that are regulated by the EPA are trihalomethanes, haloacetic acids, chlorate, and bromate. These compounds have been consistently associated with urinary tract cancers and birth defects. So as a part of this study, we conducted interviews and had meetings with community members to help design the study. We decided to look at the spatial and temporal variation of these contaminants in the drinking water system. Because we had to visit individual homes, this was a perfect opportunity for community participatory research, and it also put the local community on equal footing with other stakeholders because they could have their own scientific study. So the study is still ongoing, but preliminarily we found that there's much more temporal variation than there is spatial variation. So we see much higher levels of DBPs during the summertime than we do during the wintertime. In fact, during the summer, as many as 30% of the homes, or more than 20% of the homes, have DBP levels that are over the EPA water quality guidelines, and we found coliform bacteria in some of the homes. So we're trying to gain more funding to expand this community participatory model to other communities so that we can develop mitigation strategies to reduce exposure to these harmful compounds. Thank you.
Of the top 5 crops grown today, which one is native to North America? Soybeans, wheat, alfalfa, corn, or grain sorghum? All right, so it is corn. Soybeans originated in Southeast Asia, and wheat originated in Turkey. Alfalfa originated in Southwest Asia, which they believe was actually Iran. And grain sorghum originated in northeastern Africa, with earliest evidence suggesting it arose in Egypt. Today, each farmer produces food and fiber for how many people on average? 140, 155, 165, or 200? So I now realize this is slightly dated. In 2010, it was 155. The average is probably now closer to 200 since it's 2019. So we do have a slight schedule change. Dr. McNear had a meeting and he didn't think he was going to make it back for his slot. So Elena Jacobs, who is a PhD student with Dr. Rebecca McCauley, will substitute.
Think back to the last time you enjoyed a big, juicy steak. That steak was brought to you by gut microbes in the rumen of a cow. Now, what's interesting is that not all of these microbes are created equal. In fact, the rumen is a competitive environment of more than 1 trillion different microbes. The microbes that play by the rules make protein directly available to the animals. However, there are some bacteria that they take dietary protein and they convert it directly to ammonia. And this is not a good process because the ammonia cannot be absorbed by the animal. What happens to that ammonia is it is processed through the animal and excreted as a waste product. So NH3 is a product, or ammonia, that can volatilize in animal waste, and it can be excreted into our environments. So you're probably wondering, what does the microbial warfare system have to do with me? Well, there are 2 things. First, we need to increase our efficiency of animal production. By the year 2050, it's estimated that meat demand will rise by over 73%. In addition, we need to improve air quality for human health. The World Health Organization estimated that 7 million people died as a result of air pollution in the year 2012. So when ammonia is deposited with livestock waste on soil or in pastures or in feedlots, that ammonia can become part of the atmosphere through the process of volatilization. And once it's in the atmosphere, it can bond with other volatile compounds to create particulate matter. And particulate matter can travel long distances, and it's particularly hazardous for human health. It's hazardous to our lungs. So how do we solve these issues, and how do we meet these challenges? And meet these challenges. A team of ARS scientists discovered a new compound. This compound is naturally present in red clover, and the compound is called biocannon A. And what these scientists found was that adding biocannon A to cattle diet increased average daily gain of steers. And the reason this works is because those chemicals that are conferred through biocannon A are keeping those microbes that don't follow the rules down. So the rumen gut microbes that don't play by the rules are being affected by biocannon A. And I will be part of an interdisciplinary team that is working with lambs to feed them biocannon A and see what happens to their gains. I will be using soil to incubate manure and urine coming from these 2 trials and seeing if we can really decrease the amount of ammonia in our atmosphere. So if biocannon A can help us reach our animal production and our environmental goals, we could win the battle for agricultural sustainability. And we could do it 1 rumen at a time. Thank you.
What crop is the world's leading provider of protein and oil? Corn, soybeans, canola, or sunflower? All right, I think we agreed on that one. It is soybeans. Even though soybeans are only 20% oil on a dry basis, they are the largest source of edible oil, accounting for 50% of total global oilseed production. Which cereal grain is used the most in food items? Rye, oats, barley, or wheat?
I'll ask Google the next time I Google trivia. It is wheat, and I have no fun fact for that. Our next speaker is Ryan Collins. He's a master's student with Dr. Erin Haramoto.
All right, welcome everyone today. I'm going to talk to you about my master's thesis project I'm doing under the direction of Dr. Erin Haramoto. We mainly deal with a weed species named mare's tail, or also known as horseweed. Scientific name is Canyza canadensis. Here on the left, you can see a nice photo of mare's tail in its rosette stage. This is shortly after dormancy is broke and germination occurs. And then in the middle, we will see a nice picture of me posing with some mare's tail plants that I was tracking in the field. This is right before seed set and flowering. And on the right, we're going to introduce a way that we are we're looking to provide alternative means, or at least see the role that cover cropping has into weed management strategies. And why is mare's tail important? So mare's tail is the #1 most problematic weed in no-till soybean systems. And here in Kentucky, we are a heavy no-till state. So bottom line of farmers is the reason that mare's tail is mainly important. So why is this affecting the farmer's bottom line? Well, resistance is the main reason for this. Shortly after the adoption of glyphosate-resistant crops, shortly after, glyphosate-resistant weeds starting emerging, and mare's tail is one of those. There's also other modes of action that mare's tail is now not affected, or excuse me. There are also some other chemistry that mare's tail is resistant to in addition to glyphosate as well. So what we are doing, we are running a field project where we are incorporating cover crops with some of these herbicide chemistries to see what role they provide to farmers in terms of weed management. There are other obvious benefits farmers have from cover crops, and I'm sure everyone in here is aware of those, such as nutrient cycling, reduction of erosion. There are many more. But what we are doing, we are running a field, like I said, we're running a field study where we're incorporating these strategies with synthetic herbicides and seeing how we can provide farmers with an alternative means of keeping resistant populations down and essentially maybe reducing an herbicide application in general. So why is mare's tail a problem? Because mare's tail overwinters, and you see overwinters at this stage in Kentucky, and by the time farmers are getting in there with the herbicide program in the spring, they're already at a size they can't control. And so this is leading to a lot of the resistance problems and growing resistance problem. And we're hoping to provide a row of cover crops to reduce some of these resistance problems and giving farmers another alternative measure. All right, thank you.
How many classes of wheat are there? 2, 4, 6, or 8? There are 6. There are hard red winter, hard red spring, soft red winter, soft white, hard white, and Durham. How many kernels are present on an average ear of corn? 600, 800, 1,000, or 1,200? So 800.
The number of rows will usually be even in a normal environment because the first kernel initials split to form 2 rows from 1. I hope that's correct because that's what I Googled. A hybrid's genetics is instrumental in determining the potential number of rows, and environmental factors have a lesser effect. Our next speaker is Jimin Shin. She's a postdoctoral scholar in the seed biology lab.
Hello, I'm Jimin, a postdoc from Kawashima Lab and the Linyuan Lab of KTRDC. Today I will talk about liquid endosperm development. It is one of the early events by double fertilization of a flowering plant. How this early event can affect entire seed development. Here we can visualize what is actually happening in a super tiny seed using microscopic technique and the fluorescence marker. So we are using Arabidopsis as a model plant, and this is a snapshot of the live imaging, confocal live imaging in the early seed Arabidopsis plant. Flowering plants undergo double fertilization, resulting in embryo and endosperm. So endosperm, interestingly, they are early endosperm is nucleotide. There's no cell wall. First endosperm forms surrounding embryo, rapidly divide, and finally numerous nuclei fill in the entire cytoplasm of a seed. So we call they are liquid endosperm. Central part is the most active region where endosperm is divided, with affecting movement, affecting dynamics, the nuclei migrate toward and finally aggregate in this opposite end. So at some point, their migration and division is gradually stopped, and they start to make a cell wall. This cellulose endosperm is absorbed by embryo as their nutrient, and until this embryo is growing mature seed. Depending on how this early liquid endosperm stage is finished, how much is delayed, the final seed size and the seed quality is changed. What genes are regulating this liquid endosperm development? We can isolate the genes directly from endosperm. Most interesting thing is several hundred of genes are expressed specifically and highly in each liquid endosperm subregion. Transcription factor is it can be key as a regulated liquid endosperm and the downstream the genes. So if we can identify the transcription factor in each different subregion, we will figure out the different function of dividing and migrating endosperm in each part. Everyday people is consuming a lot of seed for their food and the medicine and different things. We can control this major factor regulating early endosperm development. So finally, we will achieve promotion of seed production. Thank you.
Which of the following soil orders are formed under forested vegetative cover? Alphasol and entasol, mollisol and spodosol, entasol and mollisol, or alphasol and spodosol? They are alphasol and spodosol. Alphasols are formed under deciduous forests, whereas spodosols are formed under coniferous forests. Alphasols account for 10% of ice-free land, whereas spodosols account for only 4%. What subordinate distinction is given for an alluvial accumulation of silicate clay? X, T, O, or H? And I'm sorry they don't match the letters. It is T, according to Dr. Google. T was for ton, which was derived from a Germanic word, which was tun, used to designate a large urn or cask, which they generally made of clay. So I don't know if you should believe it or not, but we use T. The next speaker is Dr. Tim Phillips.
Over the years, when people ask me what my job is, when I say I'm the grass breeder at the University of Kentucky, usually the next question is about their lawn. And I say, not that kind of grass. I breed grasses for cows to eat. And then they lose interest and go talk to somebody else. But the traits that are important in forage grass breeding, and specifically tall fescue, we want improved persistence, so longevity, and since animals are eating them, we want them to be palatable so the animals actually enjoy eating them and they want to eat more of them and gain more weight. And the performance is sort of integrated both with the yield and the digestibility, so there are multiple traits that are important. And the reason there's a tall fescue breeding program at the University of Kentucky is because this is the origin of Kentucky 31 tall fescue. It was discovered in 1931, and it took 12 years of research before it was officially released as a new variety in 1943. So it's been around a long time, and it's still probably 80 or 90% of the tall fescue. Several million acres, probably 20 to 25 million acres in the southeastern US. And a lot of it has been there since the 40s and 50s and 60s. So unfortunately, Kentucky 31 excels in the persistence area, partly because it has a symbiotic endophyte, a fungus that produces alkaloids that give it insect resistance and drought tolerance and anti-herbivory. It keeps animals from eating it. It makes cattle sick. So if you've ever seen cows standing in a pond in the water in the summertime, it's because they're being slightly poisoned by the alkaloids produced by the fungus that live in tall fescue. So we work with endophyte-free tall fescue. The grass can survive fairly well without the fungus, but more recently, a novel endophyte, grasses, strains of the fungus that come from related tall fescues that don't produce the most damaging alkaloid, the ergot alkaloids. So a few years ago, we released Max-Q, lace-filled Max-Q2, the first of the novel endophytes from Kentucky. It was selected for a better seedling vigor and winter hardiness in Oregon for higher seed yield. It's shown a little better disease resistance than Kentucky 31. Good drought tolerance, even this year with the late summer drought. Animals have done well, and it's persisted well under grazing pressure. So I think the future will maybe we'll forget about Kentucky 31 and lace-filled Max-Q2. I wish lace-filled was a bigger font than Max-Q2, but I'm not the one that's marketing it. So thank you.
What U.S. holiday is the top food holiday in terms of money spent? 4th of July, Thanksgiving, Christmas, or Easter? It is 4th of July, surprisingly. Americans spend $16.9 billion on 4th of July cookouts annually. How many crops in the U.S. are listed as GMOs? 6, 8, 10, or 12? It is 10. These are it, and I don't have a fun fact. Our next speaker is supposed to be Dr. McNear. He did not make it back from a meeting, I'm guessing, because he's not here. So 2 more rounds of questions. Kentucky is a biodiversity hotspot for what aquatic organism? Mussels, frogs, minnows, or darters? Of the 297 species and subspecies of mussels in the United States, 103 are native to Kentucky, and that is Monsey the Mussel Man of Kentucky. How many years does it take to form 1 inch of topsoil? 100, 500, 1,000, or 5,000? It's 1,000 years, but apparently we also need to debate moisture content and topography and organisms and all of our soil forming factors. How many minutes do we have? 2 minutes. So this made my father-in-law laugh. It may make you all laugh. So there's an old farmer, and he's walking down through his field, and he's not really paying attention, thinking about other things, and he sees a frog in the ditch. And the frog says, "Well, hello, fella." And he stops and thinks, "Oh my gosh, that frog's talking to me." And so he leans over and listens. The frog says, "Well, if you just give me a big old kiss, I'll turn into a beautiful farmer's wife for you. You will love it. You will love it." And the farmer leans over, looks at him, picks him up, and puts him in his pocket without saying a word. And he starts walking home. And the frog says, "Hey, hey, 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 why don't you kiss me and I'll become a farmer's wife?" And the guy says, "Yeah, I heard you. But at my age, I'd rather have a talking frog." What do we got? 320. 320. All right, we're going to move on a minute and a half early. So our next speaker is Dr. Todd Pfeiffer.
Begin with the end in mind. That was one habit in a popular self-help book, 7 Habits of Highly Effective People. Perhaps it's a habit that will help me in my research. As a sweet sorghum agronomist, I conduct field research, testing experimental hypotheses. What end do I have in mind? If my hypothesis is really true, I want to declare that it is significantly different than the current standard practice. So I should plan an experiment that has a relative chance of success. What does that mean? I want to have a power that's probably greater than 0.8. Power comes from how many times I repeat measurements on my data. I have to ask the question then, how many reps do I need?
How many environments do I need? How in the world am I going to decide? Well, I could use a power analysis. This graph was copied from an agronomy journal article, not on sweet sorghum, but it shows what a power analysis can tell you. So, how many reps and environments do I need? Follow the curved lines as they cross the dashed dotted line. If you follow the curved line of X's, you'll see that this experiment recommends 8 replications in 4 environments. Or if you follow the squares, it recommends 5 replications in 5 environments. Wow, that's a whole lot of work. Do I want to put that much effort into one experiment? But maybe the more important question is, what if I just stuck with the normal and I did 4 reps in 2 locations? Well, look how low the probability of success is. I'd probably be better off not even doing that experiment. The goal of my project is to summarize some of my decades of sweet sorghum data on sweet sorghum biomass, sweet sorghum juice percentage, sweet sorghum bricks or sugar content, to say, how can we do experiments that raise the total sugar percentage produced per hectare by sweet sorghum? Well, begin with the end in mind. Conduct a successful agronomic experiment. What could make it more successful? Run a power analysis first. So I wish for you that you adopt this habit of a highly effective researcher much sooner in your career than I did, and happy research.
Rothamsted is home to one of the longest agricultural experiment stations in the world. In what country is it located? Scotland, Northern Ireland, Wales, or England? It is England, and it was founded in 1843. Dr. Haramoto is researching mare's tail. What type of weed is mare's tail in Kentucky? A winter annual, a summer annual, or a winter or summer annual? It is winter or summer annual. I do not have a fun fact, so I hope you attended her seminar earlier this semester for all the fun facts. Our next speaker is Sanjay Yoshi. He's a candidate with Dr. Sharon.
Everyone in this room might have imagined or thought about how it will be without air or water. Some even cannot imagine a day without smartphones. But have you ever tried to imagine what it will be without seeds? Let's imagine, take a picture of a day without seeds. From morning, there will be no coffee, bread, cornflakes. And if you want to drive to your office using biofuel, there will be not available. And there will be no chicken or rice in the lunch menu. And if you want to relax your evening with beer or whiskey, it will be not available either. And of course, there will be no comfortable cotton pajamas to wear in the evening. So you can imagine how miserable and sad is a day without seeds. And in fact, 70% of our diet directly or indirectly comes from the seeds. Having said the importance and realizing its value in daily life and being part of seed biology group in Plant and Soil Science Department, Professor Sharon Perry's lab, where I'm a graduate student, we investigate the regulatory networks involved in seed development, specifically focusing on embryo development. After the fertilization of male and female gamete, embryo starts developing. Here on the left-hand side is Arabidopsis embryo growing in different stages. it is embedded in the maternal tissue, whereas on the right-hand side are artificially produced somatic embryos from the somatic cells in our lab using some plant hormone chemicals. So previous studies in the lab have shown that AGL15, which is a transcription factor, that means it's bind to DNA and regulate other genes, have been showed an important role in somatic embryos. However, it is also have important role with the regulatory genes involved in the in embryogenesis. But LBD40, which is my research topic, has not been studied yet. So the hypothesis of my research is that LBD40 has a role in somatic embryogenesis. In fact, the graph here shows that when there is mutation of the gene LBD40, there is low somatic embryos compared to when there is overexpression of LBD40 gene. So the objective of my research is to know where LBD40 binds in the genome and what are other genes it upregulate or downregulate. Since it's a transcription factor, which means it will bind to a DNA. And the overall, the research from this study is to be included in the applied crops, because from the basic science, we want to move to the applied science where we can apply the founding from the basic level and then using the soybean crops or the wheat, which the increasing demand of population, demand of food with the increasing population in the future. So nobody has to worry about seeds in the future. Thank you.
Who is considered to be the father of modern agriculture? Cyrus McCormick, John Deere, Eli Whitney, or Jethro Tull? It is Cyrus McCormick. He invented the mechanical reaper. He founded the McCormick Harvesting Machine Company, which became part of the International Harvester Company in 1902. Which crop below is one of the founder crops of agriculture? Chickpeas, rice, corn, or sorghum? It is chickpeas. There are 8 founder crops. They all formed the basis of the modern agriculture and arose in the Fertile Crescent between 1,000 and 10,000 or 11,000 and 10,000 years ago. They are all annual, self-pollinated, and native to the Fertile Crescent. That is our last question of the day. If you would please get a green and a blue slip of paper and vote for the people you would like to win. I'm gonna set myself as your background. Yes. Did I get it? Do we go third, second, first? All right, call people from the lobby in. I got a microphone. I don't know why I'm yelling. I was like, is this it? We're missing adults who can do things. No, it's questionable. All right, so before we announce the winners, just a reminder, we have the 3MT after party probably starting at 4:30 at Goodfellas in the Distillery District. And if you're interested, both volleyball teams are playing tonight starting at 8:40 at Marika's.
Yes, of course we'll win.
All right, our third place winner is Mohammad Fateh Ali.
Keegan, come on.
I thought I was thinking like a group photo.
No. Okay, you're done. Be a cool mom, like in Mean Girls. Do it correctly.
Our second place winner is Stuart Lichtenberg.
Work it, guys. Work it. Show your good side.
All right, Keegan. So.
Pose. Strike a pose.
Our first place winner is Dan Quinn.
I got everybody's chance.
Our student winner of the People's Choice Award is Sanjay Joshi.
One more. Wow. Amazing. This is going in Sears catalog.
And the winner of the Faculty and Staff People's Choice Award is Rebecca Ramsey. Yeah. Let me switch. Yes, you are done.
Do you need like all winners?
Yeah, we are adjourned. Oh, if you want your